Hi, everyone. Super excited to be back recording another really interesting episode after a short hiatus. We're really keen to reintroduce a steady cadence of great conversations with microbiome leaders and experts over the course of 2026. And today's conversation with Professor David MacIntyre is a great way to get started again. Professor MacIntyre is an expert in the maternal microbiome and the female reproductive tract. And what we explored today is that there's a unique ecosystem that sits inside that anatomy that has an important role to play in the health of newborn babies and outcomes during pregnancy. What you can expect to learn is what a normal microbiome looks like down there, how it can change, how it has an impact on preterm births, and what therapeutic strategies are being developed to be able to change the microbiome in the female reproductive tract to improve outcomes for mums and improve outcomes for babies. So this is super exciting. Given that we're restarting, I'd really appreciate some feedback on some of the content that you'd like to hear as the listeners. We've got a great list of experts lined up over the course of the next 6 months, but listener feedback is what really helps shape the content to make sure that it's highest possible yield. So with that, I'd like to allow you to enjoy the rest of this episode. Thank you for supporting Inside Matters. Let's make 2026 our best year yet.
So David, thanks so much for coming on to Inside Matters. I'm really looking forward to this conversation. We've really focused on the gut microbiome over the last few years. And we've talked lots about diversity being really important, the composition and makeup of the gut microbiome. But I wonder if maybe we're missing something here because there's microbiomes in other parts of the body. And if you could maybe take us back to when you had the realization that the microbiome within the reproductive tract was important. What triggered that? And why should we be excited about the reproductive tract microbiome?
Well, let me start by thanking you for inviting me to chat to about this. I mean, you're right. This realization, though, has come late to others. But many of us who have particularly worked in women's health and in reproduction have recognized microbes as being very important influences of reproductive health. My own personal story as to how I got involved in this field, though, was coming at it from the point of view of a researcher who was studying premature birth. And we recognized quite early on, and we being many people that came before me, that women who were delivering babies early would often be found to have pathogens in the lower reproductive tract. Work back in the 80s was done in and around that space using very classical microbiological techniques. And you could see that women around the time of delivery who were having these babies born too soon, if a swab was taken, you could culture out pathogenic bacteria. And of course, these were the bugs that were easy to grow, things like E. coli and group B strep and things. But as many have seen in other niches in the body, as we've started to have changes in the technologies and methodologies that we've used to characterize microbes in different niches, it became far more interesting. So around the time that the Human Microbiome Project data sets started to come out, I'd arrived quite recently at Imperial. I was working with very well-known obstetrician, Professor Phil Bennett. And we were looking at some of these data sets, realizing that many of the bugs that are often reported to be associated with preterm birth didn't seem to be the types of bugs that anyone had characterized in any of these early microbiome studies of these different microbial niches across the body. And we suspected we're probably missing a lot of those things. And those studies, which were performed by some key researchers in our field, people like Jacques Revel and Larry Forney and others, were really, I think, the first best insights that there was something really interesting worth studying. My sort of focus was more towards how that reproductive tract microbiome might be influenced or be influencing pregnancy and pregnancy outcomes. But that's where I think a lot of us started to gain our interest in this space. Really, what is there? And maybe these bugs are not just present in a sense of causing problems, but they could also be there having a very protective role. And you also mentioned diversity. That's something you probably already thinking the conversation will go down that route, which is the case that
Absolutely.
Obviously Yeah, in the gut world, it's very commonly suggested that having a high diversity gut microbiota is something that's a good hallmark of health. And typically in the reproductive tract, it's the precise opposite of that.
Yeah. That's so interesting. So one of the concepts we talk about so much on this podcast is that indeed for the gut microbiome, the ecological diversity seems to be potentially a biomarker or at least a proxy for a healthy ecosystem, and in turn is frequently associated with just a healthy state, but also improved outcomes to various different treatment strategies. And when I was digging into this a little bit before we spoke today, it became clear that, as you say, the opposite is perhaps true. So for the female reproductive tract microbiome, it's actually at low diversity, which is the biomarker. So maybe you could just unpack that a little bit for the listener. How do we know that? How do we study it? What's known at the moment in terms of the state of the art?
Sure. So as I mentioned, some of the earlier work that had been done around the time that the Human Microbiome Project datasets came out, they were the first to sort of apply next generation sequence technologies. Those studies were done in women of reproductive age who were healthy, asymptomatic, and they were sort of using those types of approaches that we all now use very commonly to examine the genomes of these microbes or specific regions that we would amplify up and sequence. And I think that was one of the first interesting findings of those data sets, which were, if you compare the diversity across all of these microbial niches, the reproductive tract really does stand out in its lack of diversity in healthy asymptomatic women. As those studies sort of started to move in towards studies of women who have pathophysiology or adverse outcomes, or maybe sexually transmitted infections or other types of adverse reproductive health, it was quite often the case that you would see higher diversity communities associating with those poorer health states. So what we know, and it's something that's actually been recognized for more than a century, is that a hallmark of good reproductive tract health is having essentially just one or two species of bacilli rods in there, which we now know to be Lactobacillus species. These were first actually described by a German physician, Dodeline, who was actually studying using microscopy at the time, potential causes of postpartum sepsis. And he realized that as he would look down a microscope in vaginal secretions, women who would go on to have a healthy outcome after pregnancy and not suffer from sepsis tended to have a lot of these bacilli rods. He very humbly described them at the time and named them as Dodeline's bacillus. But what we now recognize them to be are Lactobacillus species. And these newer molecular-based characterization of those species tells us that most women are carrying one of around 3 or 4 main species. The two probably most important or most well-known or most characterized are a species called Lactobacillus crispatus and another called Lactobacillus iners. And it is probably important at this stage to say that, while we have looked across various different methodologies, be it culture, be it microscopy, gram staining, that is a pretty consistent finding that women who are healthy are carrying these low diversity microbial communities in the reproductive tract that are typically dominated by just one of these Lactobacillus species. That being said, there are healthy women, many healthy women out there who actually do have slightly higher diversity communities. And so I think something that we may end up discussing is host response to these different types of microbial communities, probably really what the key driver is here. But broadly speaking, it is fair to say that a hallmark of health, no matter how you look at it, using molecular techniques or cultural microscopy, a low diversity lactobacillus dominated microbiome is a hallmark of health in reproductive age women.
Yeah, I'm super excited to dig into the host response, but just before we go there, when we talk about the gut, we talk about things like colonization resistance and how this broad diversity confers that colonization resistance. It's almost like a barrier to potential pathogens. When we think about the vaginal microbiome, what functions does it perform? And how does the Lactobacillus crispatus, or how does that low diversity ecosystem sort of deliver that effect?
Yeah, so there's obviously been a really important and quite different coevolution between microbes that colonize the lower reproductive tract and the host compared to, say, what's happened in the gut niche or even the skin or oral microbiome. One of the probably most important features that we think confer protection in the niche that's provided by these Lactobacillus species in particular is their capacity to metabolize food sources like glycogen and some other complex sugars that are readily available in cervical vaginal secretions and also in the epithelial cell wall, and convert that into lactic acid. And so as they do that, that produces a very low or acidic environment. They can thrive in that environment. They're very happy to be growing in a pH of 4 or even less. And what then we tend to find is that type of environment tends to be quite hostile for other microbes that may potentially get access to that niche. So other types of potentially pathogenic bacteria don't particularly like to grow well in that niche. Now, the other mechanisms that these microbes are utilizing to help prevent pathogen infection and colonization are things like competitive competitive advantages or approaches. So things like when they are dominating out the niche, they essentially totally cover the mucosal interface, which means that adhesion of some of these pathogens that might be arriving there through various mechanisms don't have an opportunity to adhere and attach and then colonize out. They're actually able to produce as well antimicrobial compounds themselves. Yeah. So things like bacteriocins, very similar to other niches, but these have been, co-evolving in a way to target some of these other pathogenic bacteria that might invade the niche.
So it sounds like they're perfectly suited for that specific part of the body, basically, and that's how it works.
Yeah, it is. But it's interesting to think that, and perhaps a little known fact for those folk who are not working in this space is that we as humans are the only species, though, that have a lactobacillus dominated reproductive tract. So our close primate relatives don't have these dominated lactobacillus niches. So it begs the question: why has that happened? That must have been a relatively recent sort of co-evolution event. That's something that my group here actually in Adelaide is now digging into, trying to understand why that's happened.
Yeah, any thoughts on why that's the case, that it's just so uniquely human?
It's tricky to say. I think there's several different pressures that these microbes are facing as to why they may have developed a very quick acceptance of that niche, and that obviously partly is because it confers benefit to the host, but it's also, I touched upon this just in the introduction there, that we should always remember that not all women around the world are in healthy states entirely dominated by these microbes. For example, if we move to African populations or Asian populations, we often see that they and it will come back to host response. They tend to have the capacity to be a bit more immunologically tolerating of some of these high diversity communities.
Interesting.
We're not really sure why that is the case. We suspect it may have something to do with exposure events around the time of delivery. Obviously, in the gut field, we know that those very early seeding events in the neonatal gut are so important for developing out the immune system and discriminating, self from a commensal from a pathogen. And so it may well be that over time, different populations have developed these types of sort of capacity to tolerate certain microbial communities. We're still really looking into that. It's a bit of an unknown area of the field.
And is this this kind of key finding associated with the lactobacillus, in particular Lactobacillus crispatus, is that common across, women who are in Australia, UK, and then also Far East and different populations like Sub-Saharan Africa? Is that consistent, or is that also, I guess, a key difference perhaps between that niche and then the gut? Whereas with the gut, the question I love to ask everyone is, well, what's a healthy gut microbiome? And then all the guests start sweating and going, oh gosh, it's a tough question to answer. But that seems to be pretty interesting that it's consistent if that's the case?
So
Do
what
we
I
know
can
that?
say that it's a great question. What is consistently shown is that Lactobacillus crispatus in particular is associated with optimal or good health states, and that's pretty much universal. What's intriguing, though, is that the prevalence of Lactobacillus crispatus is not equal across all populations. So we will see, for example, in European and Northern American populations, particularly Caucasian women, very high prevalence of Lactobacillus crispatus in healthy women. But in certain populations and studies that have been done in certain African populations, in particular, Lactobacillus crispatus may only be found in 10%, 15% of that population. So the way in which these different Lactobacillus species are interacting with host populations does seem to differ. But we do know that Lactobacillus crispatus, I don't think there's any cases of it being associated with any form of adverse health state or infectious etiology. So there clearly has some benefit for that particular species.
Yeah. So interesting, because we did get asked, I can't see who it is, but someone asked our company if we could work with Lactobacillus crispatus. And in particular, I think they were thinking about, this being used as a next generation probiotic. So it's something I'd like to get into. And there's another question I really want to ask you. I was going to wait until then, but I'll ask you now. It's totally off piste, but I just have to ask. So there's all these supplements that you can buy that you take orally, and they say that they have a potential protective or beneficial effect on the vaginal microbiome. Now I've always questioned, well, how do they find their way into the female reproductive tract if you're consuming them orally? But is there any evidence for orally administered probiotics potentially being beneficial? And if so, kind of how? What's the thinking there?
Another good question. There's been a lot of studies now that have tried to examine the potential health benefits of orally delivered probiotics that are designed really to modulate the vaginal microbiome. I think it's fair to say that there's pretty weak evidence that an oral administrative route will lead to good engraftment of the probiotic that has been taken orally. There are some lines of evidence that occasionally these microbes can make it through the digestive tract and via various mechanisms of transfer make their way into the reproductive tract. But it's not a very efficient way of getting these bugs there, that's for sure. And the other thing to think about is that the vast majority of those products, particularly the ones that you can buy off the shelf, have been developed from Lactobacillus species and strains that have had a long use in the food industry or other in other contexts, and these are not indigenous commensals of the female reproductive tract. A great example of that is Plantarum or Acidophilus. These are strains that yes, occasionally you can find in the reproductive tract, but they definitely would not be considered to be the key microbes or Lactobacillus species that we recognize as being keystone species of this niche. Now, this is not to say that taking oral probiotics has no beneficial health to reproductive tract health. I think there are ways in which potentially some of these species may modulate gut microbe host interactions that could therefore have an indirect impact on some aspects of the biochemistry and microbiology of the lower reproductive tract. But I think overwhelmingly the evidence is if you can administer these probiotics directly to the vagina, then they tend to colonize much better. And we've, of course, been involved in one of those studies recently and performed the first in-pregnancy study of live biotherapeutic. Wow. Yeah, in particular, Lactobacillus crispatus. Yeah.
And is that data available, or is that ongoing?
That is, yeah. No, so we've actually published already the safety and tolerance results of that study. So this was a study called the FLIP-1 trial. It was a product called Lactin V, which is produced by a company, Ocell, from California. And for full disclosure, I have along with many of my colleagues, a specific use patent for that specific strain for the prevention of preterm birth. But be that as it may, we did that first ever in pregnancy study, primarily focusing on whether or not that vaginal delivery of a Lactobacillus crispata strain would be safe, would be well tolerated, particularly in women who are at high risk of preterm birth and in that pregnant state. Alongside that, we of course looked at clinical outcomes. We also looked at a huge amount of work in the microbiology and immunological responses to those bugs. We've got some really tantalizing signals that this is a really interesting potential therapeutic intervention to use, and we haven't gotten to that stage of the conversation yet as to why
I know I jumped us ahead. Sorry. Yeah, I got too excited when I'm talking about probiotics.
We can loop back around and talk about that. But what I can say is that it does seem to engraft very well in pregnancy and colonize well. And that data is actually in the process of being submitted. But we've presented that in public domain before, and it most certainly seems to be very safe, very well tolerated by women who are taking this vaginally. And a tantalizing bit of information I was going to share with you is that as we published, women who were part of that trial had preterm birth rates that were quite lower than what the background expected population rates of preterm birth would be. So it does look like it is exciting. A potential route for us to explore. Yeah,
exciting. I think establishing, just from a drug development perspective, establishing a robust safety and tolerability profile in that specific population, I think, is a really big deal. That's totally exciting
particularly
for
given the fact that Yeah, it is. I mean, this is also something that many of us in the women's health field harp on a lot about. But unfortunately, particularly pregnant women are often excluded from studies, as you'll be aware. And so I think it's really important that if we're wanting to develop therapeutic interventions and help improve health outcomes in this type of space, we need to do the studies on the relevant population. So we're really happy to be pushing this ahead.
That's great. So I'll for sure come back to different mechanisms of the therapeutic avenues and so on. But I just wanted to go back to the host response. I think that's really interesting. So what do we understand then about differential host response to what's there, basically? Because that's another key element, isn't it? There's what's there and then what's happening in the body in response to it.
Yeah, it is. It's really interesting to look into, because what we find is women who are carrying these lactobacillus dominant microbial communities typically, broad brush strokes, have a very low amount of local inflammation. And we have a pretty good understanding of how that's achieved. The lactic acid produced by these species that we talked about before, we know that this is actually an anti-inflammatory compound in that niche. So lactic acid, we've got some data that we'll be publishing on this soon, and others in several other contexts have shown that if you expose, for example, vaginal epithelial cells to physiologically relevant concentrations of lactic acid, this will dampen down and suppress pro-inflammatory pathways and the production of chemokines and cytokines from these types of epithelial cells. So that's one way in which we think we're helping to improve health there, because when we have that low amount of inflammation, that also helps maintain a stronger epithelial barrier. So very much similar to the gut scenario, where if you've got inflammation present and you end up with a leaky gut, well, that increases your chances of then having infection. And exactly the same concept here in the reproductive tract. So that maintenance of that mucosal barrier is really quite critical. Then we also know that if those lactobacillus species are lost, we have then other pathogenic bacteria coming into the niche. They're not producing lactic acid, so the pH of the environment tends to go up. They then start to produce things like short chain fatty acids. And I've got to be careful the way in which I describe this. Yeah, but mostly a slightly different kind of outcome here, where I think it's very well established that short chain fatty acids have an anti-inflammatory role in the gut, and are considered to be a metabolic signature of health. Broadly speaking, not all, but many of the short chain fatty acids that are produced by these types of pathogenic bacteria in the female reproductive tract actually have a pro-inflammatory association.
Really?
So, yeah. Oh. So again, we're highlighting here just how different this local nuances exist, between microbe-host communities. Now, this all becomes very, very important, particularly in pregnancy. But before we even talk about the pregnancy scenario, you can imagine that if you're carrying a lactobacillus dominant community, which is producing lots of lactic acid, pH is low, and we might have then a virus exposure event or another pathogen exposure event. If you have a strong mucosal barrier, you're also producing a chemical microenvironment that those pathogens or viruses don't particularly enjoy. The other thing too that lactic acid does is modulate the viscosity of the actual mucus itself. So all of these things combined provide that type of innate and adaptive immune environment that's pro-protection and helping to prevent a potential pathogen infection. So as that is lost, we then have potentially this breakdown of the mucosal barrier. Mucus becomes less viscous, and viral particles have been shown to then be able to get access to the epithelial cells and beyond that. And that's when we tend to see an increased risk of STI infections. And those types of interrelationships between microbial host interactions that are pro-inflammatory or anti-inflammatory tend to hold across a lot of different pathogenic scenarios or pathophysiologies in women's health. So cervical cancer is another example. We've done quite a lot of work in showing that HPV infection followed by pre-cervical cancer, and the development from pre-cervical cancer into full-blown cervical cancer seems to be potentiated by that pro-inflammatory, higher diversity microbial environment, and suppressed by the low diversity, low inflammatory environment.
What are the specific kind of key triggers for moving perhaps from a kind of healthy state to dysbiotic state in this niche? That's interesting to learn more.
There are your obvious candidates. So some sort of an exogenous factor like an antibiotic exposure will clearly potentially, and often it's the unintended consequence of antibiotic treatment that might be designed for a gut target or something like that, or an infection in the chest. But the off-target impact is that actually the lower reproductive tract microbial communities are disturbed by this. So that we've also shown, just to highlight how clinically relevant that can be. Some work that we've done looking at women who come into a stage of their pregnancy where they might present with what's called premature rupture of fetal membranes, or when a woman's waters break prematurely in her pregnancy. We've shown that if the first line of treatment that is given to a lot of those women, which is high dose oral erythromycin, well, if you do have an infectious etiology and a pathogen present, it does a great job as expected in dealing with that. But if you're carrying lactobacillus species in your reproductive tract, it also does a fantastic job at entirely obliterating those commensals. And that might have then some further longer term negative impacts that were unintended. There's a lot of other potential factors here in play as well. So things like sexual intercourse, we think is a major source of seeding of pathogenic bacteria. Some really beautiful work actually that has been produced by a fellow Australian, Kath Bradshaw, and her team in Melbourne have recently, I think, demonstrated really nicely the importance of partner-to-partner seeding and thinking a bit more about that as a source of these pathogenic bacteria. So they've shown in a study that was published in New England Journal recently that if you treat women who have what's called bacterial vaginosis, which is probably the most common vaginal sort of infectious etiology, it's microbiologically characterized by a loss of all those healthy lactobacillus species and an overgrowth of all of these potential pathogenic bacteria. Very hard to treat. The gold standard, if you like, is metronidazole. And what they've been able to show is that if you treat a male partner simultaneously, then the efficacy of that metronidazole treatment regime is substantially improved, indicating that there is certainly some
could be the source. Yeah, it could be
at least
a source.
one potential source. Yeah.
Yeah.
Now, there's a lot of other potential sources and disturbances to that finely tuned microbial community in the lower reproductive tract that we think is susceptible to causing some of those changes. So, for example, hygiene practices like douching, which is not recommended, can change the microenvironment. We think probably there's often very low amounts of these pathogenic bacteria hidden in various different nooks and crannies of the mucosa, and they're very
Interesting.
opportunistic. So if there is a loss of pH or an increase in pH or a bleeding event, for example, during menstruation, which then all of a sudden changes the food sources for the different bacteria that might be present there, that might be just enough for them to gain the advantage in the niche and to start to colonize out and tip the balance. That's exactly right.
Yeah, that's super interesting. So maybe I could bring us back to then the preterm birth story.
Yeah.
What specifically is it, do you think, that is driving that cascade of processes, and how does it kind of start there? What do we understand? Because preterm birth is, I'm going way back now to medical school days, and it is my understanding anyways, is a significant unmet medical need still. It's around the world, it's still a really big problem, and I think the understanding of why and how it happens is still developing. So how could the microbiome in that area be important for preterm birth?
So preterm birth remains probably the major obstetric dilemma that nobody's been able to really make major headway into. Rates of preterm birth, people are just not aware of how common it is. That 10% of all births are preterm. And obviously most babies that are born preterm are born close to term. But a lot of data has been produced to show that probably even 1 day of difference in gestational age at birth makes a difference to various different parameters. When we're looking at the role of the microbiome and particularly in infections that might potentiate the cause of a baby being born too soon, we think it's more likely to be associated with the earlier preterm births. So you can divide preterm birth into a couple of different levels of extremity. If you're born, for example, before 28 weeks of gestation, then this is considered to be an extremely preterm baby. If you're born before 32 weeks, very preterm. And as you're getting later and later, and you finally sort of hit that magic mark of 37 weeks of gestation, which is considered to be term beyond that, you can see that there's this a stronger relationship between an infection or an infectious etiology and preterm birth in those earlier preterm deliveries. And that's something that's been recognized for a very, very long time. And for me to explain how the microbiome might be modulating or mediating or playing a role in that, we have to go back to your early medical school days and think about what causes a baby to be delivered in the first case, which, funnily enough, is still a bit of an enigma. We know, of course, that there are endocrine and hormonal mediating pathways that are regulating the development of the baby, but they're also regulating the uterus and how it responds to various different signals from the baby and from both the neonatal and maternal endocrine systems. But at some stage close to the end of pregnancy, what happens is that there's an activation of inflammation. And that inflammation is we describe it as being sterile inflammation. It does involve, of course, the immune system as well. But at this stage in healthy term deliveries that happen normally, there's no pathogen, there's no infection that's triggering this. This is being triggered by signals from baby to mum. And in fact, what we know that happens...
Are they just a quick, the signals you're talking about, are they sort of hormonal? Are they... They're okay. They're
hormonal. They're potentially also... There's some good evidence in animal models, not such strong evidence in humans, that they could also be, though, factors, for example, proteins that are produced by the lungs of the baby called surfactant proteins are actually ligands for toll-like receptors. So you can imagine that, and this has been demonstrated quite nicely in mice models, for example, that as you reach a certain level of maturity in utero, you start to produce a certain amount of surfactant protein, which is, of course, being breathed in and out of the amniotic fluid that's going into the lungs of the baby and then back into the amniotic sac. And then when that reaches a certain concentration, it probably is starting to bind to and activate toll-like receptors that are expressed in high levels in the amnion and other gestational tissues. That is potentially one of the several pathways that lead to an activation of inflammation. Another thing that will lead to a sterile activation of inflammation, if you like, is stretch. So some really nice work from my colleagues at Imperial, Mark Johnson, Phil Bennett, and others several years ago showed that as the fetal membranes and the uterus as a whole expand and stretch to accommodate this fastly growing baby, it does seem to reach a threshold where then you'll activate these inflammatory pathways. Now, the interconnection here with infection was that these inflammatory pathways are all regulated at a tissue and a cell level by pro-inflammatory transcription factors like NF-kappa B and activated protein 1 and others. Now, if that all happens in a healthy pregnancy towards the end of pregnancy, those pathways lead to the production of key proteins that are involved in the three sorts of events that have to happen for a baby to be born. This involves the membranes that the baby's sitting in, this amniotic sac has to rupture. Okay, that's similar to the waters breaking. And the cervix, the cervix, of course, has to dilate to allow the baby's head to come through the birth canal. And that requires a lot of tissue remodeling. So proteases and elastases and various other enzymes that are involved in those processes are regulated by these transcription factors. Those transcription factors, of course, are regulating chemokines and cytokines, which are involved in then further recruiting white blood cells into these different areas of the reproductive tract, which further helps this tissue remodeling to occur. The other thing that has to happen beyond the membranes rupturing and the cervix opening up and dilating is that the uterus has to start to contract. And for me, still, I feel the uterus has got to be one of the most amazing organs that exist.
100%.
It does this incredible job of slowly expanding and to facilitate this growing baby. And then, like clockwork, will start to contract with such force that it will push, several kilos out of a very small hole. Yeah. So that also, it turns out, those same transcription factors regulate the key pro-contractile and pro-laboring proteins that are involved in getting the uterus to contract. So to complete the story, you can imagine that if you then had earlier on in pregnancy an infection or a pathogen present, that activates an expected maternal immune response. Yeah. You then get the inflammation. And one of the tricky things is that once inflammation starts, particularly in human pregnancy, it's very difficult, if not impossible, to turn it off. And so where the microbiome comes in is we think of it as being an additional fine-tuning mechanism to help dampen inflammation or potential sources of inflammation up until you get to a point in pregnancy where, well, it doesn't really matter if there was a little bit of inflammation. Now the baby's going to come out either way. And so some of our earlier work in our lab was actually doing the first studies looking at what happens to the microbiome longitudinally in healthy pregnancy. And some other studies had been done looking at what happens cross-sectionally across pregnancy. And what the totality of that data showed us is that as you become pregnant, there is this further reduction in diversity of the microbiome, where you have really a further stronger push towards lactobacillus dominance, which would make sense with this idea of it being protective. And then that sort of reaches a peak of low diversity as you get into sort of the second trimester of pregnancy. And then as you get towards the end of pregnancy, a little bit more diversity seems to be tolerated. And I think it probably is because we're getting to that stage where not only hormonal changes, but the drivers of preventing inflammation start to be released a little bit, because ultimately you do need the baby to be delivered.
So it is, because we do this test, don't we, for group B streptococcus, in some or all, I can't remember exactly, but is that maybe then an opportunity to do more, insofar as you could maybe screen that sample for the presence or the absence of, the correct lactobacillus strains, and then maybe you look at diversity, and then you could maybe identify if someone's at higher risk at that stage? Or is that am I jumping way too far ahead then with the thinking
No,
there?
I think that concept of using a sample like a swab that is used for testing for Group B strep is a really good one. In that instance, Group B strep testing tends to be done towards the end of pregnancy, because really what matters is the potential of a baby being exposed to Group B strep. Now, the reality is that many women will be carrying Group B strep. Most women, in fact, will be carrying Group B strep at some stage of their pregnancy. It's one of those types of bugs which will be typically present in very, very low levels. It might flare and appear a little bit and then go away. And we see that when we look at studies that are specifically designed to assess and detect Group B strep in a longitudinal manner throughout pregnancy. Now, the other thing is that Group B strep isn't one of those bugs that tends to really drive the inflammatory pathways from a maternal point of view that would then cause preterm birth. If Group B strep is hanging around, though, when particularly a premature baby is born, that could be particularly dangerous for a baby. So that's why we have those detection, the screening procedures and treatment regimes for Group B strep. But the concept that you raise is a really good one, which is perhaps if right at the beginning of pregnancy and maybe some particular periods of time during pregnancy, if we had the capacity to rapidly and relatively non-invasively assess what's going on with the microbial composition, and as we've just discussed, the host immunological responses to that microbial composition, then this may be a really useful way of monitoring and observing and trying to detect women who might have a shift in their microbiome that could associate with an increased risk of preterm birth.
Yeah, because what I had noted down was could microbiome profiling eventually become a routine screening tool in pregnancy?
Yeah, I really do think it could, and it needs to be done with any of these types of screening tools with really strong evidence behind it, and probably carefully thinking about the populations that would benefit best from that type of screening procedure. And there has been a lot of effort now actually to start to investigate from a diagnostic point of view whether or not the microbiome could be a useful tool for predicting adverse pregnancy outcomes, not just preterm birth, but there has been a lot of work done in preterm birth. And excitingly, we are seeing that when we put together data sets from all over the world, really, we can see that there are some clear microbial signatures that are detectable quite early on in pregnancy that appear to be quite predictive of a risk profile associated with preterm birth. So, in fact, a lot of the work that we've done at Imperial College in this space has been funded by a US-based charity called the March of Dimes, and one of their initiatives was to fund a program of work called the Dream Challenge, where they had some researchers collate a lot of the microbiome data that's been generated out in pregnancy, and it was using a type of citizen science type of an approach, where that collated data was harmonized and that was made available to anyone and everyone who was into data modeling and predictive modeling. And they were challenged to take that data set and to develop and train a predictive model that was then validated and tested against a never before seen unpublished data set. And that showed, I think, quite convincingly that there definitely seems to be some predictive capacity sort of harbored within the microbiome during pregnancy that will give us some insight as to whether someone might be at risk of preterm birth. Now, I might just preface all that by saying as well, though, that preterm birth can be caused by lots of different things. And that's one of the challenges that we're faced in reducing rates of preterm birth. I mentioned before that we've kind of got this global rate of preterm birth of around 10%. That hasn't changed in decades. In many countries, in fact, it's gone up. And I think one of the reasons for that is that we could have a microbial component that we estimate to maybe drive 40-50% of cases of preterm birth, particularly the earlier preterm birth cases. But then there's a whole bunch of other scenarios and clinical indications and mechanisms that could be driving other cases of preterm birth that are beyond our conversation today. But a major thing that's kept the field back, though, is not having a good method of detecting and predicting women who are at risk of preterm birth. There are some tools that are used clinically. They tend to be used quite late on in pregnancy. Coming back to our earlier discussion about inflammatory pathways being switched on and being very, very hard to turn them off, typically those tools are detecting women who are likely to deliver within a very short window of time, when arguably it's too late to intervene and prevent that from happening. So one of the things I'm most excited about is this idea that if we could potentially, even before somebody becomes pregnant, optimize their microbiome in a way that we're setting them up in a scenario that at least helps minimize their risk profile, and that would need to involve some sort of tools for detection of risk, alongside hopefully a therapeutic intervention.
Yeah. So let's go there then. So what is your vision then for how we could maybe today start to do some of that, and then what's the vision for the future? What are the tools to screen and profile, and then what are the tools to maybe do the optimization?
So we've talked a bit about some tools that can be used to, of course, characterize the microbiota composition. We've got increasingly impressive, next generation approaches and molecular tools where we can actually now quite quickly and cheaply look at the composition of the microbiota. I think at the level of granularity that we would want to have that type of data collected, I still would suggest that it's not quite point of care. We're not at that stage where these tests are available at point of care. But the key thing that I think those types of tools that are really looking at composition of microbiota miss is of course host response. So there are other things that we could do, like cytokine profiling or chemokine profiling. Again, time consuming, tends to be quite difficult to have robust systems in place to do all of those things collectively in a high throughput way that we could actually action in a clinical scenario. So that actually led us to start to look at how we might address that, because I do think that if we had those two pieces of information readily available to inform clinical decisions, we could actually start to either potentially identify women at risk, put them through higher surveillance, think about therapeutic interventions, which we'll come to. And one of the ways in which we approached that was with some colleagues at Imperial, in particular Zoltan Takats, who's very well known for inventing the iKnife, which is a tool that's uses essentially the metabolic profile of tissue that's being cut through for that information, which is captured in real time, can help direct a surgeon to perhaps stop cutting through the margins of a cancer that they're wanting to resect. So working with Zoltan, we came up with a concept that perhaps we could take one of these simple swabs that we would normally go and extract and do all this type of work on, or use for a smear and a gram stain. We could apply that type of metabolic profiling, which uses a form of mass spectrometry called ambient mass spectrometry, where we generate directly from a swab detailed chemical profiles. We get an insight very, very quickly into what types of metabolites and chemicals are being produced or present in that swab. Mm-hmm. And then the concept was, well, maybe we'd be able to use that almost like a barcode of which microbes may be present. And perhaps with a bit of luck, those same sorts of chemicals could even predict the host response. And so we've done quite a bit of work in that space, which is looking really promising.
So are you maybe more excited about the potential of, I guess, the repertoire of functional activity that comes from those bugs, or you think the bugs are maybe going to be where we're targeting the screening in the future? What's your sense of what might be higher yield?
Yeah, it's a really good question. I don't think it's possible to say one or the other is better. I think if you can capture both pieces of information, that's the ideal scenario. Because I think what we are finding, and we have some other studies that we're going to be getting out there soon, where we've looked at even within the same individual, we're finding that depending on things like the hormonal environment that these microbes and host immune cells are sitting in, then the actual action and function of those host immune cells and their response to the exact same microbial community within the same host can be influenced by the hormone presence or absence, can be influenced by other factors, whether or not, for example, there's presence or absence of blood or other substrates. And so I think you kind of do need those two pieces of the puzzle, composition plus the host response at that particular given time, I think is the key for those types of screening and diagnostic approaches. And so this method that we've developed, which is called direct swab profiling the shortened form is DESI-MS, desorption electrospray ionization mass spectrometry. Simple thing is swab into a machine. Within less than a minute, we have a chemical profile that we can map to a database to work out what bugs are present and whether or not there's a certain immunological response with those bugs. That seems to provide us with a type of information that we'd want to be able to use. And then all of a sudden, we move into the second phase of the vision.
Oh, sorry, can I just ask you,
Please, yeah.
just before we get to this, is there this, I guess it's called kind of dark matter, but maybe that's the wrong phraseology. So how much do we know about what the microbes are producing within the niche? And is there, when you do some of the untargeted analysis, is there like, wow, there's a lot of stuff there that we didn't know about? Or I guess, is the field quite sure that this is generally speaking the kind of range of metabolites and outputs from the ecosystem?
There is a tremendous amount of metabolic dark matter in this niche in particular. Much like what's happened in other microbial niches, the more and more we get sensitive assays and we can dig deeper into, separating out various different compounds and detect them at lower and lower levels of detection, we realize, wow, we're missing a lot of really important information here. So there has been quite a bit of work done looking at metabolic outputs and the metabolome of, for example, cervical vaginal fluid and how the microbes might be influencing that. I think it is fair to say that microbes themselves are probably the biggest influencer on shaping the metabolic profile of the mucosal interface there. But we certainly do not have very good understanding of what those metabolites are doing from a functional point of view. We definitely know that some of them, the easier ones to detect, the ones that we can set up nice in vitro assays to examine and study, are certainly capable and do modulate some immune responses from certain types of in certain types of scenarios. Others might be involved in allowing bugs to have a certain substrate required for them to move through the cell phase in a particular way, or to adhere to another cell type, which is important for them to develop their space in the niche. There's a whole bunch of different mechanisms which are likely to be metabolite mediated. But that's, you mentioned dark matter. The other element of all of this too, which is to zoom out a little bit more, is that most of the work we've been doing in this space is very much focused on the bacterial component of the microbiome.
Oh yeah,
and we are missing.
How could we forget?
Those little things called viruses and fungi and archaea are almost certainly there.
Yeah.
We know they are there. They're almost certainly, though, also playing an important role in shaping a lot of the dynamic interactions that we're interested in. And that's something that, again, we and others are interested in exploring now as techniques become much more useful for investigating those types of microbial communities. Yeah.
Just one final point, and then we'll move on to the therapeutics. So the bacterial diversity seems to be much lower in this niche than the gut. But is the virome, the microbiome, the other microbial inhabitants, is it a similar story, or is there maybe something different going on there based on what we understand?
Yeah, it really, that I don't think we have clear. That type of comparative assessment across niches, that just hasn't been done to the extent that would want it to have been done. They're there. We know they're there. There has been some effort to start to characterize out some of the viral components and fungal components of the vaginal microbiota. We haven't even delved into bacteriophages either as another modulating factor. We could go down a very deep rabbit hole there. That is ripe for the picking. All you microbiome folk out there who are focused on the gut, I strongly encourage you, and particularly those who are working in those other spaces, come and ply your trade over on our side of the fence. And in fact, that's one of the reasons why I used to love and still love going to microbiome meetings. It's funny, when I started to work in this space 15 odd years ago, I would be typically invited to give a talk as the token, that's right, there are other microbial niches that exist. Yeah.
Another one, yeah.
More and more so, though, I think the microbiome is shaping women's health, and reproductive tract
100%.
becomes a really prominent player here. And we
100%.
are, really, Yeah, I'm really excited to see a lot of people who maybe started their career in other microbial niches, or even in totally other, microbial ecology in non-human niches and environments, are super interested and applying their trade now to the women's health space, which is so exciting for us to see.
Yeah, no, thanks so much for sharing that. I would love to talk about the bacteriophages and the dark matter, but I'm worried we won't get to the therapeutic side, which I'm super keen to get to. And I also was very keen to talk about the transfer of the microbiome from mum to baby. So maybe those two are kind of themes to just focus on in the latter part of the conversation. So you were talking about this vision, which is exciting. We've got these tools, profile the bacteria, we profile their metabolites, we create barcodes, they match the databases, and maybe we can quite quickly determine, is this person at high risk of something or not? And then maybe if they are, that leads us into the second part of the vision, which is, how do we change things?
Yeah.
So maybe we could just focus on that. That would be interesting.
We touched, I think, at the beginning of the conversation on some strategies that have been used. We've dealt with oral probiotics. I think it's fair to say that that's probably not the most effective way to directly get the right types of microbes to the niche, but bearing in mind that it is still potentially have some sort of health benefit. Now, I also mentioned that we were involved in this trial looking at directly delivering to the vagina using a vaginal applicator, Lactobacillus crispata strains, and that's producing some really exciting results. So we were talking a little bit earlier on about how we've done some work looking at directly delivering vaginal probiotics, or live biotherapeutics, as they're often referred to the niche. And I think some of the data that we've been producing is really exciting, indicates that we can get engraftment there. And off the basis of those results, we've managed to get some funding actually to start a new phase 2 study for this product, Lactin V. This is going to be kicked off very, very soon in multiple sites across the UK, funded primarily by March of Dimes and also some philanthropy backing there as well. That's really exciting. That's unlike our first trial, which was just safety and tolerance. This is a placebo-controlled, randomized controlled trial, empowered to try to see whether or not we will get a genuine reduction in preterm birth rates. So that will be, I think, the key trial to keep an eye on in this area. There has been quite a bit of work outside of pregnancy looking at this product as an example.
Sorry, can I just a little bit some more questions? How big is the study?
Sure.
When do you expect it to start? And what are the key endpoints that you're excited about? We talked about, is it a primary endpoint? Is like true efficacy assessment, or you still need to build some safety and tolerability data? How is it looking?
Yeah, so the primary endpoints will be a reduction of preterm birth. That's what the study design is. So we're pushing the boat out. We think that's really what we want to get an answer as to whether or not this is a viable way forward to help mums and babies in this state, and that's the right way to do it. As part of that work, of course, we will be doing a lot of additional fun science, looking at host responses to these colonization dynamics. We'll be looking at also other relevant secondary outcomes that will be important here. So that will include looking at some of the early outcomes of babies. We'll probably get onto this discussion. We're interested to know whether or not that type of treatment regime in pregnancy may have implications for baby during the time of delivery, for example. Do some of these microbes that colonize the reproductive tract genuinely transfer over into the neonatal gut microbiome, where they may have an important function or role? So this is really exciting to be part of, and I think you'll see quite a bit of a few announcements around that trial quite soon.
Yeah, it's an exciting time to be in our space, because
it really is. Yeah, it is. Yeah,
I guess you could say, 7-10 years ago, we weren't doing these statistically powered definitive studies, so to speak. So it sounds like it is very exciting because we talked about the importance of Lactobacillus crispatus. We talked about all this kind of observational evidence, basically the breadcrumbs, and you're kind of pulling the story together. Now this study is going to actually, hopefully, prove that these early results have not occurred by chance, and then there you go. We might have an intervention now, which that's huge.
That's exactly right. I mean, and I think for someone who's been working in this area now for quite some time, one way or another, we will advance the field from a trial like this. We can either see that the type of result that we may have inferred from earlier results where we can make a difference, and that's obviously incredibly exciting. But even if we don't, being able to definitively have that data that tells us that that's an avenue we shouldn't be exploring, or we need to adjust, various aspects of dosing regimes or whatever it is, we will make some major steps forward in the field from the results of this trial. And that's for someone, as I say, who's been involved in this for a long time, that's where you want to be. You want to be doing that type of definitive study to get an answer. Some people I've worked with and colleagues have said, oh, what if it doesn't work? Well, for me, even if it doesn't work, we're going to learn so much. Exactly.
That's drug development, right? It's binary. It's binary because it has to be binary. And of course, people are going to say, what if it doesn't work? Well, yeah, but what if it does work?
Yep.
And that's the question we're trying to answer here. If it does work, then think about all that potential benefit that could come in years, decades to come, where we've actually proven that this has a benefit. So I'm waxing lyrical now about drug development. So this obviously is key, and it's very exciting. And congrats on getting the funding. It's not easy. So it sounds like everything's coming together there, which is great. I had a note to ask you about, because one of the other guests did actually mention this, vaginal microbiome transplantation. Because we've spoken so much about fecal microbiota transplantation on this podcast. Like, it is that a thing?
Yeah, it is a thing. It is a thing. So there's a couple of different areas to cover in that conversation. So the first of those is to think about some of the concepts we've already covered, which is that if we start at the most basic concept of a probiotic treatment, where we're placing typically one strain of a particular species into a niche, and we're trying to create an environment where it will engraft and colonize out, it may well be the case that that's not sufficient for the types of host responses that are required, to really support engraftment and to see the functional responses we want to see. And that's led to some folk to ask that question as to whether or not, well, maybe it's not just the microbe itself. It might be more broadly the microbial community and everything that's within it, and the microbiome, as in the metabolites that go along with a transplant of, if we wants to take a vaginal secretion and transplant it into another individual, you wouldn't be just transplanting the microbial component. You would be transplanting as well a lot of substrates that go along with it, other elements that may be critical here. And so there has been studies that have looked into testing the feasibility of that. I think it's very early days. Those studies, which have been published in very important journals like Nature Medicine, but have been really done in small patient numbers. So there's an important study done by an Israeli group looking at around 5 women who had recurrent BV, and the early data indicates that some of those women do appear to respond quite well to that as a process of treatment and transplanting the entire vaginal secretion into the niche. There are, I think, several issues around regulatory considerations, feasibility, scale up, that make that as an approach really quite challenging. But most of the folk, I think, who are thinking about that as a strategy, it's more of a proof of principle, and then working out whether or not we can identify maybe a synthetically determined community composition that may be supplemented or complemented by other factors like certain metabolites or other factors that enable an improved response when those types of more complex products would be developed out. There's some good rationale for that, I think, in the sense that when we look deeper into the data, we've talked about Lactobacillus crispatus, for example. Well, not all Lactobacillus crispatus are probably the same. When we go down to strain level, there are multiple strains of Lactobacillus crispatus that will have functional potential that will be perhaps better in some individuals than others. And so that's led to some consortia and research groups looking at developing out multi-strain or multi-species combinations of these types of probiotic therapies. There I think are still some features that lend itself to, particularly from, you know this yourself from working in your company, there's issues around scalability, and when you start to move then to combining multiple species, that it becomes a real challenge to produce that at scale at the type of quality and expected from TGA and MHRA and all these various other regulatory bodies that expect that. So
100%.
There will probably need to be a balance there to what the ideal combination of features will be. But this concept of vaginal microbiome transplant is a thing. It's happening and it's being tested. And then, of course, we have this concept around the time of delivery, which may be where this conversation goes to now, which is that baby, as it's being born through the birth canal, has really its first major dose or seeding event right then and there. And so there've been several studies now to indicate that babies that are born via cesarean section have quite a different early life gut microbiome than those that are born vaginally. There's a concept that is perhaps because they're not being exposed to vaginal microbes. And that has led to some proponents of vaginal seeding of babies, which is, I think it's still fair to say it's relatively controversial. There's a lot of effort that's been put into, particularly by the groups who are trying to do this at scale and to test really the feasibility and safety of this, where there are pretty good procedures in place to screen carefully the microbiota before exposing that to baby, bearing in mind too that if baby would have been delivered vaginally, baby would have been exposed to these microbes anyway. But that is some. Yeah, go ahead. Sorry.
Interesting, relevant, recent story. I shared the great news that my wife and I had 6 months ago with our.
Yeah, congratulations again. Fantastic.
Thank you. I appreciate that, and it was really quite interesting because, when our son was born by cesarean, it was originally planned cesarean. And I asked, I said, so, is there the ability to ask for seeding? And also, what's the position within the hospital and seeding? What's best practice? They'd never done it before, and they asked me what it was. They didn't know, and it wasn't something that we'd actually done much of at all. Now I'd only done a very limited amount of reading just to understand, first of all, is it done within the NHS, and also kind of what the best, not best practice, but what the current consensus was around whether it was a good thing or not. But it was just interesting that actually they were doing very many of these procedures every day. But within the obstetricians that I was speaking to, no one was really kind of thinking about it as a potential avenue. So it feels like maybe in the UK it's quite early in terms of thinking. Have you got any thoughts on?
I think that's fair to say that. Yeah, I mean, a lot of that. The first studies in that space came out 6-7 years ago, and there was a very strong backlash, particularly in the UK, against it. And I think quite rightly at that stage that there wasn't good data to indicate that it is a safe practice, and particularly considering trying to minimize risk. If a baby's being born via cesarean, and we know that baby won't be exposed to the vaginal secretions of the mother, then the onus would be on us to very carefully confirm that if we are going to take microbes from the vagina and expose it to baby, we want to be really sure that there's no, for example, group B strep or any other pathogens present in that sample. And so I think at that stage, that particularly in the UK and some of the other Commonwealth countries that follow similar guidelines, there was, generally speaking, not strong encouragement for that process to be continued. I think a lot of mums and dads and families are still very interested in the concept of it, and a lot of my clinical colleagues still tell me that there's a lot of interest in it, similar to your situation, where it's often raised by the parents as something that maybe ought to be considered. But overseas, it's certainly gaining traction, and I suspect it won't be the end of the story. There are several larger studies that have been done recently, particularly a major collaboration with some folks in New York and researchers in China, where they've done a very carefully conducted trial and shown that babies who were exposed following cesarean section to the mother's vaginal secretions tended to have better longer-term outcomes in sort of the 2-year range from measurements of neurodevelopment and some other allergy measurements. They tended to have better outcomes than babies that were not exposed to mum's secretions. Now, we need a lot more data before we'd ever be able to encourage or recommend that type of practice. But I find it intriguing, just the concept that we have evolved to have that dosing event at the time of vaginal delivery. Typically, the way in which evolution works is that we don't usually have leftover interactions like this that are meaningless. So I'm still pretty convinced that that exposure event does have some sort of important role. There have been some really, really high quality studies done looking at the transfer of strains from the reproductive tract into babies' neonatal gut microbiota. That they're really challenging studies to be done. Technically, those studies have been done phenomenally well. They indicate that there's probably a very small amount, if any, of mum's vaginal strains colonize in the long-term baby's gut microbiota. Yeah. However, that doesn't really surprise me. I think the types of bugs, as we've talked about today, that are colonizing the reproductive tract niche are not have not evolved to colonize the gut microbiota for very long at all. They're also in relatively very small amounts when a baby is being born. They're being exposed to small amounts. So my suspicion is that they may be important in the very, very, very first sort of pioneering seeding events, which may then help other obvious keystone species like bifidobacterial species that are coming from breast milk or skin microbiome. Yeah, that's perhaps helping them to engraft and provide some cofactors that enables them to be the sort of first major colonizers in the neonatal gut microbiome. Very hard to prove that, though.
Very hard to prove. You're also really kind of challenging my thinking on this a little bit, because it's now clicking a little bit. If it's a very low diversity environment, then it definitely is not going to be providing all the strains which eventually become the adult gut microbiome. It's more likely to come from the environment. So it's challenged my thinking there a little bit, I have to say. And are there any thoughts around, you talked about this supplement, next generation probiotic, really, that's Lactobacillus crispata. Are there any thoughts about very, very early life supplementation somehow, either in the breast milk or when they get down the line a little bit, maybe something else? Are there any thoughts on that being high yield? Or is it mainly we're thinking about pre-birth event?
No, I mean, in my area, we're focused primarily on the pre-birth scenario, but there is a lot of work being done in seeing whether or not probiotics and other prebiotics may be a useful strategy to support optimized development of the gut microbiota. That's particularly in the case of babies who are born prematurely. If you haven't had her on yet, I strongly advise you to have a chat to Lindsay Hall or Christopher Stewart. These are two people who do fantastic work in this space. Also present incredibly well. I think it's quite convincing that is a avenue that a lot of people are wanting to explore. Because it does seem to be some good benefits of getting babies exposed to those types of species in that way.
Awesome. Could I ask you, just as we kind of move towards the end, to give us your vision for where you think we'll be in 10 years' time? How's all of this going to come together, do you think? We can be optimistic.
I'm always optimistic. I do think we are really on the cusp of making some major advancements, particularly in and around adverse pregnancy events like preterm birth, where we haven't been able to make much of an inroad. And it really does move towards this concept of a personalized medicine approach or stratified medicine approach, however you want to call it. For too long in these types of fields, we've sort of considered all of these cases to be a similar type of disease state. Well, the baby's born prematurely, acknowledging that it could have reached that conclusion via various different mechanisms, but have not really offered any new tools or therapeutic interventions to stratify out on the basis of mechanism or underlying fundamental mechanism, and then an appropriate matched therapeutic to deal with that. Now, it's funny because, of course, in the cancer space, this is how all therapeutic interventions are done. There's careful screening, there's careful assessment of various different host factors, genetics, et cetera, and then the most optimized therapeutic intervention is selected, which we can clearly see has a major improved outcome on treatment efficacy, on health outcomes, etc. My vision is that that's the type of approach we'll see in the pregnancy space, and more broadly in the women's health space, where we assess carefully microbial components. We assess carefully host response to those microbial components. And on the basis of that, we're making a much more informed decision, which will help us to then select a suitable therapeutic that then can modulate those interactions that we know are so important for generating a risk profile associated with STI infection or in pregnancy, preterm birth risk, or even sepsis after pregnancy. I think that is going to be applied more generally, and probably, I suspect, in 10 years' time, we'll move away from a reactionary approach to this type of work and clinical care, where we see a symptomology and we try to do something about it, but instead have a much more thoughtful approach to it. So perhaps, as we've talked about before, it could be even in the preconception stage, where if one's thinking about becoming pregnant, then there's an effort made to optimize these types of microbe host interactions, which are likely to probably improve even conception, to improve some of the key aspects of the pregnancy, like invasion of the placental arteries into this concept of placentation, so important, and then help to prevent the early activation of inflammatory pathways, which could lead to preterm birth. And that, as we've touched on just at the end there, may set up baby really well, because baby then is more likely to be delivered into a microbial environment, if it's a vaginal delivery, that's most suitable and perhaps most supportive of then subsequent exposure and colonization of bifidobacteria and other species that sets them up for life. And that's indeed what I'd like to see, that we really get that window of reproduction optimized as much as possible, which then would have a major knock-on effect in later on in life.
Yeah, no, thanks so much for sharing that. I just sent you an email before we started saying I think this is the next big frontier, and the conversation we've had today has really cemented that for me. So I'm really glad you've come on and given the listeners an insight into this, extremely important niche that we've just not talked enough about. So, David, thank you for your enthusiasm and your expertise and insights. Really enjoyed this. If a listener wants to learn more about your work, where do they go to find more about what you're doing?
Yeah, so I've got a couple of teams that I work with still. Our team, which is the March of Dimes Prematurity Research Centre at Imperial College. You can find us online. I've got a team over here at Adelaide University, where I'm the director of the Robinson Research Institute. You can pop me an email. I'm also on LinkedIn and most of the socials, although lesser so these days. But very, very happy to talk to anyone about science.
Thanks, Dave. Really appreciate it.